Hello, everyone. Welcome back to another episode of healthXaudio. I am one of your hosts, Theodora.
And I'm your other host, Adrianne.
Yeah, so after a long hiatus, today we'll be talking about cancer from an immunologic perspective. Immunology is the study of the immune system and how the body defends us against infection and disease.
Dr. Buechler is an assistant professor at University of Toronto's Department of Immunology. His lab, just founded in 2021, works to uncover the foundational aspects of fibroblast and macrophage biology required to discover therapeutic targets.
As we have recently discovered from our brief conversation ahead of time, we've only found out that he's been here for basically only a brief period of time, as his lab was only founded in 2021. So our warm-up questions may or may not apply to him, but we'll give it a shot. So starting off with one of our favorite questions, we always like to ask, what is your favorite food truck in Toronto? U of T has a lot of food trucks. Have you tried any?
Yeah, that's a great question. So, I went to grad school in Seattle and then spent time in San Francisco. And so when I think of food trucks, I think of Portland, where there are lots of big food truck pods. And I don't know if if food truck culture is as prevalent in Toronto, but we do definitely see some on campus. So we have little kids and we've gone to one food truck. We went to Shawarma King and it was great. So that's the only one we've been to. We hope to go to more in the summer.
Yeah, that's a great one.
Yeah, I think the food trucks just aren't around because school isn't open, but I definitely love Shawarma King. I've gone there multiple times last year, so great choice.
Let me ask you guys a food truck question. What is Canada Pasta? I walk by it every day. It's never open. At Robarts, have you guys seen this food truck?
Canada Pasta.
I'm assuming it's one of those. There's a lot of small pasta restaurants, kind of chain pasta restaurants around the area where you walk in and they make you a pasta. It's like a DIY pasta bar, and I'm assuming that's what the food truck is.
Me too, but it hasn't been open the whole time since we've been here. I walk by it every day. Never been open. My kids are clamoring to eat Canada Pasta.
Maybe one day.
Yeah, I haven't seen that on campus, but I've definitely seen the restaurant version of Canada Pasta, and
Oh, okay.
Yeah, so maybe it's just because it's winter. I don't know.
Okay, so for our second warm-up question, what's been your favorite experience at U of T or in Toronto so far?
Toronto seems like a great city. It seems super livable. So I think we're enjoying the winter. So it's fun to have snow on the ground and experience all 4 seasons. But I think our favorite part has been going to splash pads in the summer, just enjoying the heat and then getting wet. We think that's been super fun. So doing that and exploring different neighborhoods has been awesome. We're really enjoying the city.
Yeah, so you mentioned that you were in Seattle beforehand. I'm also from the West Coast. I'm from Vancouver, so just a little bit up north.
Huh.
I was wondering, what do you think is the greatest difference between Seattle and Toronto?
Yeah, I think we're still getting to know the city. I think the biggest thing we miss maybe isn't the right word, but that there aren't mountains close by. That makes it feel far from home.
Mm-hmm. I agree. I used to navigate by looking out for the mountains. I'll be the mountains are there, that's north, and that's how I navigate. Yeah, it's definitely weird here. I use the CN Tower instead. That's a great one. So let's get started on the main questions that we have had prepared today. So on the topic of cancer, many lay people may have only heard of the term chemotherapy or chemo as a method to treat cancer. In the case of immunology, what are some of the major ways we can use immunology research to treat cancer?
Sure. I think the big picture for the interest in cancer immunotherapy, which has been relatively recent, is that traditionally cancer has been treated through either surgery, so removing a cancerous tissue, which was done, a century ago and is still done now. And then there was a progression towards chemotherapy where you would treat a patient with drugs that may somewhat selectively target cancerous cells, but have a lot of side effects and don't work all the time. I think what, the revolution that we're experiencing now, and I think that's evidenced by the fact that Jim Allison won the Nobel Prize a couple of years ago for cancer immunotherapy, is this concept that, maybe we can ameliorate cancer by triggering the immune system to attack cancer cells. So we all have an immune system. It's what keeps us healthy in most cases. And if we can use therapeutics to target our own existing immune cells to a cancerous cell, maybe that's a new way to combat cancer.
I recall from like a first year class that I mentioned previously, there's this type of thing that people do where they like take the blood from a cancer patient, they like look at what the cancer cells are doing and then like try to find targeted therapies or like, I guess, drug concoctions to like target what's happening in those cells. Like, I guess that's technically a chemo thing, but I don't know, like, am I wrong? Is this something we could expand upon? Yeah.
I think there are a lot of ways that, one way to think about looking at an individual patient and trying to understand what is specific about what differentiates their transformed tumor cells from a normal untransformed cell is important. So the immune system is very elegant in the fact that it's kind of designed to recognize things that shouldn't be in the body. So the kind of paradigm is set up as the immune system recognizes non-self. So if something's in your body and it's not supposed to be there, it's not part of you, it has the mechanisms to wall that off, eliminate those things from your body and to keep you healthy. The tricky thing about cancer is a transformed self. And so just by virtue of that, it kind of makes targeting cancerous cells challenging because they are at their core a self cell that you wouldn't want attacked. But these cancer cells undergo mutations that cause them to be cancerous. So if you can identify what those changes may be, you may be able to then tell your immune system, like, sure, this looks like it might check out as a self cell, but there's some non-self components to it. And therefore, we want to target those cells. And so when we think about maybe looking at individuals with a specific type of cancer, broadly as a population, we would want to identify those biomarkers that may identify a cancerous cell and enable us to potentially reprogram the immune system to attack those cancerous cells.
Really interesting. I would definitely love to see where that goes in the future. But let's move on to our next question.
Yeah, what I wanted to ask is, since your research focuses on fibroblasts, we're going to start with a few questions about that. So to begin, what are the functions of fibroblasts in normal tissue, and how are they different in cancer?
Fibroblasts are cells that exist everywhere in the body. Every one of your tissues has fibroblasts within it, and the role of a fibroblast is to generate the proteins that give tissues structure. So I think of them as defining the architecture of tissues. So they give tissues the rigidity or like literal flexibility that they need. So they're the cells that kind of make a tissue what it is. They're the structural pieces, and they're found in every tissue. So they're very important just by virtue of that. There are also some kind of add-ons that fibroblasts bring to different tissues. Research suggests that there are different subsets of fibroblasts that can take on specialized functions. So for example, in the lymph node, in addition to secreting the extracellular matrix that makes a lymph node rigid, the fibroblasts also, a subset of the fibroblasts, also secrete factors that help keep the immune cells within a lymph node happy. So they can kind of play several different roles in terms of, again, the architectural aspects, which are essential, but then also perhaps, having a more specific role in a given niche. So in the context of cancer, often, and it varies depending on the cancer, but in many cancers, you will have cancerous cells that emanate from, again, a transformed cell in the tissue. And then you oftentimes see fibroblasts encircling that tumor, kind of walling it off and to anthropomorphize what they might be doing. So they're often localized near tumor cells. And what they can do in terms of immunotherapy is that, this barrier of fibroblasts that are secreting proteins that oftentimes are used to kind of provide stiffness and matrix to a tissue can prevent the immune cells that we want to kill a tumor it may prevent them from physically engaging with those tumor cells. So T cells are a cell within the immune system that can be specific for a transformed cell, and they have the ability to kill tumor cells. But if we have a T cell that's specific for a tumor cell, either by virtue of the fact that it was generated in your body, or if it was given a therapeutic to help license it to kill tumor cells, if the T cell can't get to the tumor, it's not going to make a difference. And fibroblasts, oftentimes, as I said, surround these tumors. So we think that maybe manipulating the fibroblasts that are associated with the tumor may enable the T cells, in this case, to actually get to the tumor and eradicate it.
Yeah, that's a great answer. I like thinking about fibroblasts and all their different roles. I think in my histology class, we're often responsible for identifying different factors underneath like slides. And one trick we have is if you're not sure what something is, it's probably a fibroblast.
I love that.
Yeah, right. Am I wrong in saying, I think I seem to remember fibroblasts being involved in scarring.
Absolutely. So fibroblasts are essential players in a scar reaction, where these are the cells that when they get activated, they can be contractile and secrete these hard matrix proteins. So they're the cells that are responsible for kind of closing the wound. And, when you get a cut or a scrape, it can just kind of feel like it's not part of your ordinary skin, that's kind of, it doesn't have the same rebound that your skin does. I mean, I'm old, so my skin doesn't do what it's supposed to do anymore anyway, but all you young kids may feel that way. And when you have a scar that's healing, it's hard. And that's because the fibroblasts are contracting and secreting matrix. So in a scar, if everything works as it's supposed to, your fibroblasts get activated, they secrete the matrix, they help the scar develop, and then hopefully they go away after the scar has healed. So in the case of cancer, you often have these fibroblasts that are associated with tumor cells, and one way to describe cancer is almost like a wound that won't heal. And so you have these fibroblasts that are there all the time, being activated, being in many cases pro-tumorigenic, or helping the tumor grow.
I am no longer healing like I should as well, so I relate to that. Haven't healed like that since I was like maybe 5, but well, we'll live. At least I still clot, so that's a good thing. In regards to kind of the research being done on like removing fibroblasts from cancer tissue, like what has been done and like what are we working towards right now?
Yeah, I think that's a big question. So if the model is that fibroblasts can be bad, what you might want to do is either eliminate those cells or potentially reprogram them to be good. And we're still learning whether we can do either of those things, or, in which scenarios one of those may be the right approach. But you could envision a scenario where if you find a marker on a tumor-associated fibroblast, so some specific protein that differentiates it from a regular fibroblast, that you could maybe generate an antibody, which is a protein made by the immune system, very specific. We can also generate antibodies. We can generate them in a laboratory. So we could generate an antibody that's specific for the fibroblast, but may also have a chemotherapeutic associated with it. And so then you can have an antibody that goes to the fibroblast and kills the fibroblast to, again, open up the niche for T cells to enter potentially. Or you could have an antibody that's specific for a fibroblast and provides a signal to cause it to differentiate in a different way, maybe to reprogram it to be like one of those fibroblasts inside the lymph node that helps with immune cell localization. So I think it's all about, for now, we're trying to understand what markers are relevant to think about targeting fibroblasts, and then answering the questions of, well, are these cells, when they're in the tumor, can they be reprogrammed? Do they need to be depleted? If we can identify these specific subset of cells, we can then understand what we can do with them. But we're still at the point where we're kind of trying to delineate the activation programs of fibroblasts in different cancers or in other diseases as well. So we're still kind of writing the book as we go along. It's an exciting time to be studying fibroblasts.
That's really cool. Like I kind of find that like as a 4th-year student, like whenever I ask a question, oftentimes the answer is like, we're doing research on that now. And it's really cool to see like that this is the, I guess, like the forefront of research that we're doing right now. So another thing that your lab studies is macrophages. So we want to start off with like the basics. How do macrophages behave in normal tissue?
Sure. So macrophages, somewhat similar to fibroblasts, I consider a basic building block of tissues. So in all tissues, as I said, you have fibroblasts, you also have macrophages, and you probably have, well, you have endothelial cells, which are the cells that enable blood vessels, for example, to grow, or lymph vessels to exist. So macrophages can kind of develop in a couple of different ways, but they're kind of like garbage trucks in tissues in a lot of ways. So cells are always dying, making different byproducts. Macrophages go in, gobble those up, keep things clean, and also can play important roles during infection, where they can identify bugs that you may be infected with, and can also help prime subsequent immune response.
That is very cool. From what I used to know about macrophages before your sentence was like, it's like a more like general immune system response. They'll like see a foreign entity and try to eat it. Like that's kind of like,
Yep, they do a lot of that for sure.
yeah. In terms of like, I guess like in tumor tissues, how do macrophages act like differently?
Tumor cells can also, as I said, fibroblasts that are associated with tumors can act to impede an immune response, and fibroblasts oftentimes just physically segregate the immune cells you want to kill a tumor from the tumor itself. Macrophages can also play a suppressive role for the immune response, meaning that they can kind of slow down the kind of immune response that you might want. Both fibroblasts and macrophages are very acutely aware of their environment, and the environment that they're within really helps program what they do. And so in a tumor microenvironment, a lot of the signals that macrophages see cause them to take on kind of an immunosuppressive phenotype. So where they may be secreting factors that limit the effectiveness of T cells or make T cells less happy. Therefore, then you get less T cell based killing of your tumor cell, for example. So I think fibroblasts and macrophages often go hand in hand, whether that's in a normal tissue. So we think that fibroblasts secrete growth factors that keep macrophages happy. And on the other side of the coin, macrophages secrete growth factors that keep fibroblasts happy. So they kind of exist in this circuit. And in a tumor, that circuit is still probably alive and well. So I think they're dependent on one another, but also the macrophages themselves can acquire additional functions that may limit the effectiveness of an immune response to a tumor.
We've already kind of talked about how, like, what research we're doing on like fibroblasts and how to get them out of there, essentially. But in terms of macrophages, like, what are we targeting? Is it that like we're trying to get them out of there as well, so that they don't like upset the T cells, or is it that like we're trying to, you know, reprogram them so that they can help the T cells instead?
Yeah, I mean, I haven't quite thought about it as like that, but it's quite similar to what's going on with fibroblasts, at least from my perspective, where we're trying to identify what makes a macrophage derived from a tumor different than a macrophage in a normal tissue. And if we find something different, we can now target that with a specific treatment. So the key to cancer or any disease is that you want to have a therapeutic that's as specific as possible to that scenario. So you're not damaging normal tissues. So, I think in terms of macrophage research, we want to understand what's special about a tumor-associated macrophage. Can we find a target, so a protein that's uniquely upregulated, and then can we generate tools that can either deplete it or potentially reprogram it?
Okay, that's interesting. So this is more specific to like lab procedure, I think. Like, what are you guys doing to differentiate these things? Like, is it like X-ray crystallography where like we're trying to map it out and like looking and being like, this looks a little different, maybe we can target that? Or are there other methods that you're working with?
Sure. The approach that's most common these days, which is still relatively new, is the idea that you can isolate cells from a given tissue or a tumor, and then you can understand what the array of genes is in that tissue, and you can do it at a single cell level. So you can extract single cells, and then you can know what is the transcriptome of each cell in that tissue. And hopefully then you could identify a population of, say, macrophages. And then you can see that they express the RNA or the transcript of something unique to them relative to normal tissue macrophages, and that this may be a target. And so I think that's where a lot of the field is going. And the concept is kind of under the umbrella of single cell genomics. So we can understand what is happening in each individual cell at the time point that we're looking at them. And this powerful technique, or this kind of suite of techniques, is, I think, really unlocking lots of potential for therapeutics.
So in terms of like the single cell genome sequencing, would it be like, do you guys then like compare the genomes of lots of different cells to be like, okay, I think these are like the overlapping ones, and like maybe that's what we should target? Or is it more like just comparing like one to another and being like, what is the difference? Or maybe you guys do both. I don't know. Yeah.
Sure. So I think, for most sequence, it's all about how you set up your experiment. But typically, I think you would want to at least start by looking at, in this case, a tumor tissue versus a normal tissue. So if you're looking at the pancreas, a pancreatic tumor, and a normal pancreas. And then, you might compare, you might then identify what clusters of cells are near each other, and that those cells, looking at the gene expression of them, are likely to be macrophages. So you have macrophage from cancer, macrophage from normal tissue, and then you can say, but what's different about cancer? Macrophages from cancer, do they express something different than a macrophage? Well, maybe that would enable us to target these macrophages. But then, because we have an array of other cells from a tumor, we can see if that special target is actually unique to macrophages, or maybe other cells express it in the tumor too. So you can kind of do these comparisons on a large scale to identify truly unique targets, which is what you would like.
I'm assuming that this would be like a whole separate procedure for like each organ in the body, because lots of differences there.
Yeah, absolutely. And that's where kind of the fun is. You can see what's different about individual cell types and how that changes during cancer, early in cancer, late in cancer.
I'm glad to hear your enthusiasm about it. I want to know more about what your lab is currently working on more specifically.
Sure. So as I said, our lab started in August, or we moved here in August, and right now we're focusing primarily on fibrosis. So during fibrosis is a disease in which oftentimes the fibroblasts, which as I said, make kind of define the matrix of cells, define the architecture of cells, where these, in the case of fibrosis, the fibroblasts become activated and secrete a lot of extracellular matrix, which causes tissues to physically stop working. They just harden up. And so we're trying to understand the biology of these activated fibroblasts during fibrosis. And we think that's useful for understanding the pathogenesis and potentially therapeutics for fibrosis. But some of our work has indicated that, there may be similarities between an activated fibroblast in a fibrotic tissue and an activated fibroblast in a tumor. And so if we can identify unique themes within fibrosis, we may be able to then test them in cancer to see if there's overlap too. And so, we're interested in cancer as well, but we're starting with fibrosis. And so we typically use mouse models of fibrosis, and we're working to understand fibroblasts through a lot of the techniques we spoke about. So using single cell approaches, we also can isolate fibroblasts and look at them by flow cytometry or look at their protein expression. So it's kind of just getting started here, but we're excited about it.
Oh, wow. Y'all joined us at the start of the school year, essentially. That's super recent. Yeah. So I guess I was about to ask how far you guys were getting, and you did mention mice models. I was wondering how you're using mice models in this process. Are you kind of growing tumors and then looking at slides of the tumors and comparing, or is it also kind of a genome sequencing thing that you're doing as well?
Sure. So I think the key for us is using mice that have been specifically designed to enable us to see different fibroblasts or different macrophages, and then to specifically target these cells. So for example, if we identified a target specific to a tumor-associated fibroblast that we think is relevant to their biology, we can use some of our systems to eliminate that gene specifically from fibroblasts. And we can say, well, if we eliminate this pathway in fibroblasts, does that make tumors grow more or less? Animal models are really useful for proving a lot of the concepts you may draw. I have my misgivings about it, but it is a very powerful tool in our arsenal to, try to better understand the biology of these cells that we want to target in humans.
Mm-hmm. Do you think that in cancer research, like mice models translate really well to human like therapies and stuff?
Yeah, in some cases they can be very useful models. What you really need to understand your biology is kind of overlapping models. So you need to test things through a number of different ways. And if your answers all check out, then that means you have a really robust phenotype. And mice are certainly not perfect. Mice are mice. Obviously, we aren't mice here. The mice that we study are also genetically all the same, which is very different than humans. But because their sameness enables us to ask very tailored questions that we can potentially apply to human medicine, which is challenging. It's all about having these orthogonal or complementary techniques, and transgenic mice are just kind of one tool in our quiver that enables us to better understand cancer.
I guess this is kind of like a random question. I'm in a class currently called Sex in the Brain. It's like very much talks about like how sex differences really influence like medicine and like treatments and stuff. Like, do you think that in cancer research, like sex differentiation is like an important thing? Like, I know that since the mice are all like genetically similar, it might be a little easier to see sex differences. I guess, like, is there anything on that? Like, does it matter?
Oh, yeah, for sure. Matters a lot. Down the line, hopefully, if you have cancer, you will have a very individualized treatment program that is specific to your tumor type, your sex, your age, all kinds of this stuff. And I think before we can actually move to precision medicine on that scale, we still need a fundamental understanding of the immune system, which we're still developing. So all these things go hand in hand. I think we need to push precision medicine, but we also need to fill that in on the back end with a complete understanding of the immune system. And certainly sex plays a role in immune responses.
I feel like with a lot of scientific advancements or like medical advancements, like our goals are like there. Like we're like, we want to do precision medicine, but like we don't have enough knowledge to back it up very often. Yeah.
Yeah, absolutely. But you have to have that goal, without that, you're just kind of, you need that finish line,
Yeah, speaking about your research, one thing that really fascinated us was your Fibro Explorer, which has their same X in our name, like healthXvisuals. So very interesting.
Yeah.
Yeah. Get our lawyers in contact about that one. So
awesome.
Yeah, for the listeners, we want to let them know Fibro Explorer is an interactive data portal that allows users to examine gene expression at the single cell level in around 230,000 mouse fibroblasts from 17 different tissues, 50 data sets, and 12 disease states.
Yeah, so we generated this data explorer as part of a paper we published last year. And as you said, Adrianne, basically what we did was we took all of the single cell gene expression from fibroblasts across tissues and diseases and mice and humans, and we tried to put it together in kind of a centralized place using a web tool that anyone could go and look at the gene expression of their favorite gene in fibroblasts. And it just is a tool for folks that are interested in a particular pathway to understand if that's relevant in fibroblasts and which type of fibroblast. It's all about trying to give back to the community and to push science forward. And I think it's just easier to move science forward if everyone can see the same data. And so we hope it's been useful.
To my understanding, like Fibro Explorer has been just your data so far, or are you guys opening it up to become like a collaborative database?
Yeah, so I mean, what we did for Fibro Explorer is actually anytime you publish a paper that uses federal funds, at least in the US, I'm sure it's true in Canada as well, you need to deposit any sequencing data that you use or you generate using, taxpayer money. And so all of that's publicly available. So what we did was we took mostly published data sets and integrated them into one big data set. And that was useful for us to define some fundamental characteristics of fibroblasts. And so we released our integration of that data. Yeah, so we generated some of it, but not a lot of it. But the integration part is valuable, I think, across tissues and models.
I love that. It's like a huge meta-analysis of all these. I guess all the research that has been done already.
Yeah.
Did this take you guys a really long time? Like traditional meta-analyses may take a really long time to get through. Like, did this take you a really long time, or was it just like plugging it in?
Oh, sure. I mean, it certainly was a lot of work, and you know, I'm more of an experimental biologist, but I've worked with a lot of really talented bioinformaticists. So it was certainly a Herculean effort to get the data in the right place, clean the data, and integrate it in a way that we think reflected biology. And you know, we had some data that showed that what we did was probably right. And so, you know, I think there's huge value in computational undertakings and the application of computational tools in biological sciences. And I think the real value added is when you combine wet lab experience, so you know, doing physical experiments like I talked about with fibrosis or cancer, and then pairing that with more of the genomics approaches.
I think you already kind of touched upon like your inspiration for creating FibroXaudio. You mentioned that for educational purpose and also for like, you know, letting everybody have access to data is really important in scientific research. How do you hope like this information will be used? Like, what are you hoping other researchers will do with it?
Sure. Well, I mean, we hope that it helps enable them to fast track their research. Maybe they're interested in a particular gene, they can look if it's there, if it's not there. You know, by sharing the data, hopefully we all get further faster together versus individually. And so, you know, we hope that people use it to tailor their research programs as needed.
Yeah, I think like the collaborative nature of science is really important to us. I think that's what kind of aligns with our goals in a way, to sort of spread information as well as best as possible.
At the end of the day, yeah, we're all just walking each other home. So, you know, hopefully we can work together as much as possible.
Are there any specific topics or projects that your lab is working on that you think are really cool and interesting and you want to share?
Sure. Yeah. I mean, we're stoked about how now that we have we're developing a more complete understanding of fibroblast subtypes and kind of the differences between different fibroblasts. We're hoping to be able to identify the targets and do some of the fundamental biology that's required, whether it comes to macrophages or fibroblasts, to proceed with optimism on a specific target or pathway. And so I hope the outfall of our research is, at the end of the day, we're doing both fundamental research and translational research. So hopefully we move science forward, but also can generate therapeutics that are relevant to patients as well.
I was on your lab page. Like, your lab seems to be quite small right now. I mean, it's a starting team.
Oh yeah, no, it's just me and some other person. We have a research associate who's awesome named Afshin, and we have 2 postdocs joining. Super small.
Are you in your lab right now?
Yeah, you bet. Well, I'm in my office, but yeah, today was a big day. We just had like refrigerators and freezers delivered. So
awesome.
Big time stuff around here.
Lab setup time. We love that.
Yeah, it's so sweet.
Awesome. So thank you so much for your time today. I personally learned a lot, and I have a very mild immunology background, and I hope our listeners have learned a lot from this interview as well. Thank you for taking your time out of your, I guess, reading week. I don't know if you have a reading week to spend time with us.
No, thank you guys for taking time out of your reading week. This is when I assume you go on vacation ordinarily, but yeah, I mean, yeah, thank you, Theodora and Adrianne, and you know, I'm really supportive of, you know, your open source approach to disseminating information and making it easy for people to ingest. And so I think our philosophies align, and I think it's great. So I applaud you guys for reaching out to me and more qualified and interesting individuals.
Thank you.
Thank you.
Thanks, everyone.